Alright, hey guys, so today I'm going to be covering exam 3 material. So we're going to be going over most of what is on the exam. So for certain, you guys are going to be going over the lac operon, the tryptophan operon, as well as the gal system. You'll also have some material going over the Drosophila sex cascade. You'll have some inversions, some polyploidy, aneuploidy. Essentially, everything from Unit 2 or Unit 3 will make an appearance in the exam. Now, I would highly recommend you guys go through your recitation notes and make sure you have a thorough understanding of all the concepts. That's essentially all I have to say about the exam. Going into the actual material, let's talk about inversions. All right, so right off the bat, what is an inversion? An inversion is a segment of DNA that has been flipped 180 degrees. So if we were to have an inversion occurring between C and D, so we would have this piece of DNA cut off from both of these ends, and then it would be flipped 180 degrees. That would result in us having a new DNA sequence of ABDCE as opposed to ABCDE. This entire chunk of DNA was flipped. Now there are two types of inversions. There is a paracentric and a pericentric inversion. A paracentric inversion is an inversion that does not include the centromere. And here is an example of a paracentric in which we don't have an inversion, and in which we don't have a centromere that's being included in the inversion. Now, a pericentric, on the other hand, is an inversion that includes the centromere. And here's an example of it. Here, for this inversion, we have an inversion occurring between genes B and D. When we get the flip of 180 degrees, we have the genes swapping with one another. And now we have A, D, C, B, E, and we have the centromere in a different location as compared to before. This can have a variety of effects. So one of the effects it can have is areas around the centromere, including the centromere, are high in heterochromatin or closed chromatin. Now, genes that are near heterochromatin tend to be expressed to a lesser extent. Though they are still expressed, they tend to be expressed to a lesser extent. Now, if you were to given a problem and you were asked about both of these genotypes, if they were viable, the answer to that would be yes, because ultimately they still contain all the genes that they need, and they're still going to produce all the genes. But some effects could be that some of the genes are expressed to a higher extent or to a lesser extent. Ultimately, for this example, the centromere is still near B and C, as it is in the beginning, so we're going to have the same expression. The genes are just going to be located in different areas, but it's still going to be viable. Now, an easy way to kind of remember the difference between paracentric and pericentric is that paracentric has the word acentric. When you put A as a prefix in front of other words, that typically means not included. So you have acentric, or you have achiral, in which it's going to be not chiral. So a paracentric would be an inversion not including the centromere. Pericentric has I in it, so you can think of I as including the centromere. All right, going on to the lac operon. The lac operon is inducible, meaning that it is regulated by an inducer molecule. So the example I've given below is just an example of the lac operon, in which the lac operon, which consists of 3 genes, lac Z, lac Y, and lac A, is involved in the metabolism of lactose. The operon is normally repressed by the lac repressor protein. However, when lactose is present, it binds to the repressor protein, changing its shape, preventing it from continuing to bind to the operator, allowing RNA polymerase to then transcribe the genes that will then result in proteins to break down lactose. So ultimately, the lac operon is a set of genes meant to break down lactose. It would be energetically unfavorable to produce these genes if you have no lactose present, because the goal of these genes is to break down lactose. So when lactose is present, you're going to produce the genes to break down lactose. When lactose is not present, you will not produce the genes to break down lactose, because that would be energetically unfavorable. The lac operon contains structural and regulatory genes. Structural genes are genes that encode for RNA or proteins that are not regulatory. So examples of that would be LAC-Z, LAC-Y, and LAC-A, which encodes for beta-galactosidase, permease, and transacetylase. Essentially, structural genes are, in a way, the product of the operon. That's essentially what they are, the product of the operon. They don't have any effect on the actual operon itself. Regulatory genes, on the other hand, are genes that encode for RNA or proteins that are regulatory. And an example of that is the repressor protein. Now, I do want to mention that the repressor protein and the repressor gene is not a part of the actual operon itself. Here is an image of the operon. All this right here is the LAC operon. This includes the promoter, the operator, and all the structural genes. On the outside, we have the repressor genes. Now, though this is not a part of the operon, it still regulates the operon. For that reason, it's a regulatory gene of the operon. There are also cis-acting elements and trans-acting elements. Cis-acting elements are essentially DNA sequences that are next to the genes that they regulate. And examples of that are the promoter and the operator. So these genes, or DNA sequences, regulate all these structural genes. And another way to think about it is these are adjacent to the structural genes, and they are immovable. They are stationary. Transacting elements, on the other hand, are proteins that can bind to areas of the operon, either increasing or decreasing transcription. And an example of a transacting element would be the repressor protein. The repressor protein ultimately leads to either the ability to not break down lactose if it's binded to the operator, or the ability to allow for transcription to occur if it's not binded to the operator. And transacting elements are movable, meaning that it can go from one chromosome to another. That's what makes cis-acting elements versus transacting elements different. Transacting elements are movable, whereas cis-acting elements are stationary. All right, to go over some of the mutations that can occur, if you were to have a mutation in the promoter, that would result in RNA polymerase's inability to bind. If RNA polymerase cannot bind to the promoter, you cannot initiate transcription. So that ultimately is going to lead to repressed expression. If we were to have a mutation in the operator sequence, that would result in the repressor protein's inability to bind to it. If the repressor protein is unable to bind to the operator, there's nothing stopping RNA polymerase from transcribing the rest of the genes, and that's going to result in constitutive expression. Going on to the trans-acting elements, if we were to have IS, that results in a mutated repressor that cannot bind to lactose. So it's going to stay bound to the operator. So if the repressor is continuously binded to the operator and it's unable to bind to lactose or to change its shape and detach itself from the operator, that is going to result in repressed expression, since RNA polymerase is never going to be able to get past it. If we were to have a working repressor protein, it would be inducible, since when lactose is present, it can bind to it. When lactose is not present, it will not bind to it. I- results in a mutated repressor that cannot bind to the operator. So if the repressor protein is unable to bind to the operator regardless of if lactose is present or not, then there's nothing stopping RNA polymerase from transcribing the genes. That will result in constitutive expression. All right, so here's some practice. Beta-galactosidase, or BGAL, is the Z gene, whereas Y, or permease, is the Y gene. Let me erase these right here. All right, so let's go through these problems. Will BGAL be produced, or Z? In order to figure this out, first and foremost, you have to look at the Z genes. Let's see if we have functional Z genes. Both of these are functional, which means that we can produce beta-galactosidase from both of the genotypes. If there was a Z minus, that would mean that we would result in a non-functional beta-galactosidase protein, which means that we would not be able to produce it. Since it would be unfunctional, it would not have any purpose, so we would not include that. Now, we know that both of these are able to produce beta-galactosidase. Now, let's look at the mutations. Oh, I also forgot to mention, for the mutations, this is a hierarchy that I like to follow, in which if you have a mutation in the promoter, this is the highest of the hierarchy, meaning that any other mutation will be negated. If we have a mutated promoter and RNA polymerase is unable to bind, no transcription can occur. And if we were to have, for example, a mutated I minus or mutated repressor protein that results in constitutive expression, even if we were to have I minus, P minus would take prevalence over I minus, since RNA polymerase is unable to bind to the promoter region. So it's unable to transcribe the genes. Likewise, if we were to have a mutation in OC and we were to have a mutation in IS, which results in a super repressor, OC would take prevalence over IS. Alright, so now let's look at the mutations. Here we have a functional P+, here we have a functional O+. Looking at the other genotype, we have a P+ and O+. So these are all functional, all working. Now let's look at the transacting elements or the repressor protein. Here we have an I+, here we have an I+. So we know that this is a fully working system, which means that it's inducible. Since it's inducible, that means that when lactose is not present, we will have no production of these genes, and when lactose is present, we will have production of these genes. So we'll have no production here, we'll have production here. Going on to permease, or Y, we see that we have here we see we have a functional Y, and here we have a functional Y. All the other genes are the same, and it's going to be inducible, so that's going to result in us having the same result of no production when no lactose is present, and production when lactose is present. On to the next example. Looking at Z, here we have a functional Z, here we have non-functional Z. So right off the bat, looking for BGAL, we don't have to look at this genotype since this cannot produce a functional Z protein. Looking at the mutations, here we have P+ and OC. We have a functional P, and now here we have an OC. OC is higher on the hierarchy than everything else apart from P, so that means that this will take prevalence over all the other mutations. So OC will be expressed, so OC ultimately result in constitutive expression. So we will have constitutive expression of the Z gene. So regardless of if we have lactose present or not present, we will have constitutive expression. Going on to Y, here we have functional Y. I'll use blue for Y. And here we have a functional Y. Looking at the other genotypes, here we have a P- or P+, here we have an O+, here we have the same P+, OC. OC takes prevalence over everything else apart from P. So that will result in constitutive expression always. Now, if we were to have a P- here, we would still have constitutive expression of Y, because we have another genotype that can still, this genotype right here, can still produce Y. So it would make up for the lack in the case that we had a P- over here. Alright, for this example, looking at the Z, here we have non-functional Z, here we have a functional Z. No need to look at this genotype for Z. Looking at the mutations, here we have a P+ and an OC. OC is going to result in constitutive expression, second highest on the hierarchy. So we're always going to have, we're always going to have expression of beta-galactosidase. Going on to Y, here we have a functional Y, and here we have a functional Y. Looking at the mutations, here we have a P- and an OC. Since we have a P-, regardless of the other mutations, P- is highest on the hierarchy, which means that RNA is unable to bind to the promoter. So this entire genotype is essentially scrapped. Now the only thing you need to look out for is the repressor protein. If we were to have an IS, this protein would be able to go and take prevalence over this protein since it's higher on the hierarchy right here. However, in terms of structural genes, this will not be able to produce anything. So we don't have to look at that for Z. or for Y. Looking at the other genotype, here we have OC. Recall, OC is highest on the hierarchy, higher than all the other genes or mutations on the hierarchy, apart from P. Here we have a functional P. So OC is going to be able to produce Y constitutively. Looking at the other genotype, for Z, here we have functional Z, here we have a non-functional Z. No need to look at this for Z. Looking at the mutations, we have a functional P, functional O. Now we have to look at the transacting factors, such as the repressor proteins, which are able to move from one area or one chromosome to another. Here we have an IS, or I super repressor, and here we have an I plus. This IS is higher on the hierarchy than I plus, and that's going to result in repressed expression, as you can see here. So since we have repressed expression, we are never going to be able to produce Z. So it's going to be permanently repressed. For Y, here we have a functional Y, here we have a non-functional Y, so no need to look at this for Y. The mutations, here we have a working promoter and a working operator, so now we have to look at the transacting factors, or the I, or the repressor proteins. IS is able to move from this genotype to this genotype, so it's able to take prevalence over I plus, so that's going to result in permanently repressed expression, so we're going to have no expression of permease. All right, for this genotype, looking at Z, here we have a functional Z, here we have a functional Z. Looking at the mutations, here we have a P plus, O plus, here we have a P plus, OC. Right off the bat, oh my goodness. Okay, here we have OC. Since we have a working promoter here, OC is second highest in the hierarchy, so any other mutation doesn't matter. It's going to result in constitutive expression of Z, so we're always going to have expression of beta-galactosidase. Going on to Y, here we have functional Y, here we have a functional Y. Here we have IS, and here we have I minus. Now IS takes prevalence over I minus, however, OC takes prevalence over IS. And since this has a functioning Y protein, we're always going to get constitutive expression of permease. For this genotype, looking at Z, here we have functional Z, here I have a non-functional Z. No need to look at this genotype for Z. Looking at the mutations, we have P plus, OC. Right off the bat, since we have OC, we know that it's second highest in the hierarchy. So any other mutation apart from P minus is not going to impact it, so it's going to be constitutively expressed. So we're always going to have expression of beta-galactosidase, or Z. For Y, we have Y minus and Y plus, so we don't have to look at this genotype for Y. Looking at the mutations, here we have O plus, P plus, and we have I minus. So you'd have two I minuses. I minus results in the repressor protein's inability to bind to the operator, so that's going to result in constitutive expression, ultimately resulting in us always being able to produce permease. Onto this genotype, looking at Z, here we have functional Z, here we have a functional Z. Looking at the mutations, here we have P minus and we have O plus. Here we have OC and we have P plus. For this genotype, P minus is going to render all the rest of these genes, apart from the repressor, as useless. Whereas for this genotype, we have a working promoter and we have OC. OC is second highest, so since we have a working promoter, OC is going to be expressed. So we're always going to have expression of the Z gene. So beta-galactosidase is always going to be produced. For Y, here we have a non-functional Y, and here we do have a functional Y. However, we have a non-functional promoter, and that's going to result in the inability to produce any genes from this genotype. So we're never going to be able to produce permease. Here we have an extra example. I don't want to solve this because I already went through the other one. However, I will give you guys the answers to it. And this space right here means that this genotype goes down for it. So the answer to this would be plus plus, minus minus, minus plus, plus plus. If you have any questions, just email me about this and I can explain it to you. Alright, going on to the tryptophan operon. The tryp operon is a repressible system, meaning that it is regulated by a repressor protein. When tryptophan is present, tryptophan represses transcription from occurring. Here we have a beautiful image of the repressor protein when it's binded to the operator. So the repressor protein is going to be binded to the operator when you have tryptophan binded to the repressor. So when tryptophan here is binded to the repressor, it gives it the correct shape to be able to bind to the operator, and that represses transcription. Recall that ultimately the goal of the tryptophan operon is to produce tryptophan, the amino acid. Now, if we had tryptophan present in the environment, then it would be energetically unfavorable to produce more tryptophan. So for that reason, when tryptophan is present, it's going to bind to the repressor, causing the repressor to bind to the operator, repressing transcription, not allowing it to occur. Transcription only occurs when we are in need of tryptophan. When tryptophan is absent, the repressor protein is unable to bind to the operator sequence since it does not have the correct shape to bind to it. Now in the tryp operon, you have an added layer of regulation It's been a long day. Okay. In the tryptophan operon, you have an added layer of regulation known as the attenuator sequence. The attenuator, as I just mentioned, is an added layer of regulation that allows transcription to occur. When tryptophan is present in high levels, that results in what's known as the terminator harpin. The terminator harpin represses transcription and does not allow it to occur. When tryptophan levels are low, that results in what's known as the anti-terminator harpin. So the anti-terminator harpin allows transcription to occur. And from a conceptual standpoint, what happens is, if for some reason RNA polymerase was able to get past the repressor protein, when you have a lot of tryptophan present in the environment, to answer this question, I guess, or to explain what I want to say, you have to understand the difference in protein synthesis and gene duplication and replication in prokaryotes versus eukaryotes. Remember, operons are in prokaryotes, and the method for transcription and translation and DNA replication differs between both eukaryotes and prokaryotes. In eukaryotes, that occurs in the nucleus and is then transferred from the nucleus into the ribosome into cytoplasm. However, in prokaryotes, you don't have a nucleus. You have a nucleoid region, which is just present in the cytoplasm. So in eukaryotes, you have transcription in the nucleus and translation in the ribosome. That's located in the cytoplasm. In prokaryotes, you have transcription and translation occurring simultaneously, since it's all occurring in the cytoplasm. Now, when you have high levels of tryptophan, that results in high levels of tryptophan-charged tRNAs. And remember, tRNAs are the RNAs that carry amino acids to the ribosome, and they allow for the making of proteins. When you have a lot of tryptophan present and you have transcription occurring for the attenuator sequence, that is going to result in the ribosome being able to translate very quickly, and that results in the terminator harpin forming. Now, when the terminator harpin forms, that causes RNA polymerase to bounce off of it. So, though transcription may be able to occur in the beginning portions, as soon as it reaches the terminator harpin, it's going to bounce off, so transcription cannot continue. However, when tryptophan levels are low, that means you have low levels of charged tryptophan tRNAs. When you have lower levels of charged tryptophan tRNAs, it takes longer for them to reach the ribosome since you have less of them. So that causes the ribosome to be stalled, which allows the hairpin to form in a shape that is known as the anti-terminator shape, and that allows for transcription to occur. All right. So there's also a mutation hierarchy we have. We have R+, which results in a functional repressor, and we have RB, which is the same as R-, and that results in the repressor's inability to bind to tryptophan, resulting in constitutive expression. R+ is higher than RB or R- on the hierarchy. So here we have a practice problem. Will the TRPE enzyme be produced when tryptophan is present or not present? Will the TRPD enzyme be produced when tryptophan is present or not present? Looking at this genotype, here we have functional repressor, functional operator, functional E, and functional D, and non-functional D. So the D enzyme will never be able to be transcribed, at least in a functional manner. So we will never have D produced, so we can, right off the bat, remove that. For E, we know that this is a repressible operon, which means that when tryptophan is present, we are not going to have transcription occurring, so this will result in no transcription occurring. Whereas when no tryptophan is present, we will have the synthesis of this enzyme, so we will have this being produced, which for this, I'm going to replace those into no's. Looking at the genotypes, here we have R-, we have O+, E+, D+, we have all functional structural genes. We have a functional operator, and we have a mutated repressor. But we have a plasmid that carries a functional repressor. Now remember, repressors are transacting, meaning that they can move from one area to the other. So the repressor protein produced from this plasmid will be able to take prevalence over this non-functional repressor protein. That's a terrible arrow, but ultimately resulting in us having a fully functional genotype. Since it's fully functional, that means when tryptophan is present, we will have no expression. When tryptophan is not present, we will have expression. Likewise, same goes for TRPD enzyme. Going on to the example underneath it, here we have all functional genotypes. Here we have RB. R+ takes prevalence over RB. So it's going to be the same result as the one above, since it's repressed. No transcription when tryptophan is present. Transcription when tryptophan is not present. No, yes. For this example, we have all functional structural genes, including the operator. We have a mutated repressor, but we also have a functional repressor that's located on a plasmid, so that's going to take prevalence over the non-functional. And that's going to result in us having the same result as the answer above. No expression when tryptophan is present, and expression when tryptophan is not present. No, yes. Onto this example, here we see we have a mutated a functional E and a mutated D. We also have a mutation in the repressor protein, resulting in constitutive expression. So for the TRPE enzyme, we are always going to have the expression of the TRPE enzyme, since this will result in constitutive expression. Now for the TRPD enzyme, we know that this is non-functional, so it will never be produced. Going for this example, here we have all functional genes except for the repressor protein. This repressor protein results in constitutive expression, so we're going to have constitutive expression of all of the genes, or all of the enzymes. All right, going on to the Gauss system. The Gauss system is essentially just a set of genes in eukaryotes that breaks down galactose into glucose. Now, the Gauss system contains 4 structural genes: Gal1, Gal2, Gal7, and Gal10. And an easy way to remember this is 1 plus 2 plus 7 equals 10, as I've highlighted here. The Gauss system also has 3 regulatory aspects. You have Gal4, Gal80, and Gal3. Gal4 is a transcription factor that binds to the UAS region, and that initiates transcription. Gal80 is a repressor protein that binds to Gal4, and it prevents transcription. Gal3 is a regulatory protein that binds to galactose, allowing Gal3 to then bind to Gal80, releasing it from Gal4 and allowing transcription to occur. So Gal4 binds to the UAS region. The UAS region is just a region, it's just a DNA sequence in the promoter region, and that is an area which allows for this protein or this transcription factor to bind and initiate transcription. Now, when Gal80 is bound to Gal4, it's inactivated. However, when galactose is present, galactose then binds to Gal3, causing Gal3 to bind to Gal80, which then releases Gal80 from Gal4, activating Gal4. And now, Gal4 will be able to transcribe the genes. Here's a mutation hierarchy in which Gal4 is the highest, Gal3 is the lowest. When Gal4 has a mutation, that results in the inability to bind to the UAS region. So since Gal4 is unable to bind to the Gal4 region, it's unable to initiate transcription, and that's going to result in repressed expression. A mutation in Gal80 results in the inability to deactivate Gal4. So now Gal80 has an issue binding to Gal4 and deactivating it, keeping Gal4 activated, which results in constitutive expression. A mutation in Gal3 causes Gal3's inability to bind to galactose. So if Gal3 is unable to bind to galactose, it's going to be unable to bind to Gal80 and release it from Gal4. So Gal80 is always going to be bound to Gal80. Or Gal80 is going to always be bound to Gal4, which results in repressed expression. Now I want to mention these are all transacting, meaning that they're all movable. Here we have an example. Will the Gal structural genes be transcribed or not transcribed for each of these genotypes in growth mediums with galactose and growth medium without galactose? So here we have, so in order for an organism to grow in galactose, it has to have all functional genes. If some of the genes are unfunctional, then you will not be able to produce the proper enzymes to catalyze and break down galactose into glucose. Because ultimately that's what we're trying to do with this. Or that's what's trying to occur. So if we have all functional genotypes, we're going to be able to grow in galactose. If we have any mutation in any of the structural genes, that will result in the inability to grow in galactose, since it won't be able to break it down into glucose. Here we have an all functional genotype. So when galactose is present, it will be able to grow. And when galactose is not present, it has no medium to grow, so it will not grow. Here we have a mutation in the UAS region. So if we have a mutation in the UAS region, that means that Gal4 has a problem binding to that UAS region or sequence in the promoter, meaning that transcription won't be able to occur for the specific genes that they're asking for. So that means that since we have a mutation, we won't be able to break down galactose into glucose, so we will never be able to grow in any medium, at least containing galactose. For the example underneath here, we have a functional Gal3, functional Gal4, and we have a mutated Gal80. Gal80 is a regulatory aspect or regulatory protein. So when we have a mutation in Gal80, that results in constitutive expression. So we are always going to be able to grow in any media. Now for the example underneath it, here we have Gal3 minus and Gal80 minus, as well as Gal4 plus. We have a functional Gal4, mutated Gal3, and Gal80. On the hierarchy, Gal80 is higher than Gal3, and Gal80 results in constitutive expression, so we're always going to be having constitutive expression of these genes. Alright, going on to the Drosophila sex cascade. So the Drosophila sex cascade is just a series of steps to produce proteins with male or female specific splicing factors. So here, if we look at this model, here we have a female that's dependent on the X chromosomes to autosomal ratio. For females, you're going to have a transcription factor that will be able to bind to this sex lethal gene and transcribe the sex-lethal protein. This sex-lethal protein is going to be able to splice down this mRNA or pre-mRNA in a way that removes this stop codon, and that ultimately results in the ability to produce what's known as the transformer gene. This transformer gene is going to be able to splice this gene right to this portion, leaving in this stop codon and this gene right here. And that ultimately results in the DSXF protein, or a female-specific version of the DSX protein. For males, we have no transcription of the sex-lethal gene, and since we don't have the sex-lethal protein, that will splice in a different manner, leaving in a premature stop codon, which will not allow for the production of a transformer protein. The transformer protein will then, the absence of a transformer protein will then lead to the mRNA splicing of this specific gene, of the pre-mRNA splicing of this gene, including the stop codon, which results in a male-specific DSXM protein. Now, these proteins here are similar in structure, but they differ in activation domains. So they bind to the same regions. They have the same DNA binding domains, so they can bind to the same region. So here, if we were to have an image of the protein, we would be able to bind to the same DNA sequences, but these would be activated differently on top. You'd have to have an extra protein or some sort of factor that adds to this, activating it. So they have different activation domains. Now, ultimately, what matters is the production of these proteins. So if we were to have, for example, if we were homozygous for a mutation that removes the sex-lethal gene, so if we had no sex-lethal gene at all, that would result in us not being able to produce the sex-lethal protein, so we would not be able to splice this in a female-specific manner. That would result in us having males as a result, as the final product, I suppose. If this organism were to carry a transgene that produces a functional TRA, a transformer protein, without the sex-lethal gene, so if we were able to produce this protein right here in the absence of this sex-lethal gene, then that would result in us having a female being produced. Remember, these genes right here ultimately don't matter. What matters is we get to this final product. So if we were able to insert a transformer protein, then that would ultimately result in us having the proper splicing to produce a female-specific protein. However, yes, in this case, the one I'm circling is a male, but if we were to produce this transformer protein, or we were able to insert it somehow, that would ultimately result in female-specific splicing, as in this example right here, giving us a female-specific protein. All right, going on to epigenetics. So for epigenetics, or essentially the non-genetic gene regulation, that has two aspects to it. It has histone modifications and DNA modifications. For histone modifications, you have acetylation and deacetylation. When a histone is acetylated, you're going to be adding an acetyl group to the histone, and that results in having open chromatin, and that facilitates transcription. Now, open chromatin is the same as heterochromatin, and open chromatin is the same as euchromatin. Heterochromatin is the same as closed chromatin. So remember, open chromatin or euchromatin is going to result in us having a strand of chromatin that's loose. So that's going to allow for RNA and transcription factors and DNA replicating factors and whatnot to be able to easily access the chromatin and being able to transcribe the rest of the genes. For closed chromatin, or heterochromatin, the chromatin is going to be very tightly packed, not allowing RNA polymerase or any of transcription factors to be able to access the chromatin, not allowing for any expression of the genes. Back onto histone modifications. When you deacetylate a histone, you're removing an acetyl group, and that results in having open chromatin, and that represses transcription. That results in having closed chromatin, and that represses transcription. For DNA modification, if you have methylation and unmethylation. When DNA is unmethylated, that's the removal of a methyl group, and that results in open chromatin, and that facilitates transcription. When you have methylation, you have the addition of a methyl group, and that results in closed chromatin, and that represses transcription. So for open chromatin, DNA is unmethylated and histones are acetylated. For closed chromatin, DNA is methylated and histones are deacetylated. All right, well, that's all I have. Study hard. Hope you guys do well on the exam, and most importantly, don't stress too much about this, and be sure to subscribe. Smiley face.